name is Hibba Ahmed, and this summer I had the opportunity to work in Dr. Brown's biochemistry lab, where I studied G-protein coupled receptors, or GPCRs, which are a class of membrane proteins heavily involved in cell signaling and cell interaction. And for this reason, they are major targets for many drugs and therapies. Specifically, I looked at rhodopsin, which is a light-sensitive GPCR found in the retina that changes shape when activated by light and transitions through two intermediates, known as the pre-active META1 state and the active META2 state, which is when the signaling process begins. Many studies that look at GPCRs, such as rhodopsin, do so under artificial conditions, but I investigated how environmental factors such as temperature and hydration can affect GPCR activity to better reflect how they function in their biological settings. We used UV visible spectroscopy to track rhodopsin's activity between the two intermediates during our experiments, and the first experiment that we conducted was a Van 't Hoff to better understand the temperature dependence of the META1 META2 equilibrium, and additionally to understand how hydration can also affect rhodopsin's activation. We induced different water levels through the use of polyethylene glycols, or PEGs, of different sizes. And our results showed that with higher temperatures or higher hydration levels, the META1 META2 equilibrium was shifted furthest to the right to favor the active META2 state. Furthermore, we conducted a basis spectra to measure the amounts of META1 and META2 present under different conditions. And while our results showed that lower pHs do in fact favor the active META2 state, we also found data to suggest the existence of a potential additional intermediate beyond META1 and META2. Overall, our results showed that environmental factors such as temperature and hydration do in fact have an effect on rhodopsin's activity, and on a broader scale, this applies to not only GPCRs, but proteins in general. Thank you.
Hello, my name is Azani Anderson, and this summer I work with Alexander Bookja's Computational Plant Science Lab. The lab as a whole is focused on learning more about plants, but specifically with the focus on learning about root systems. This is important because roots are a relatively understudied part of the plant, especially compared to how important they are for the plant as a whole. It also must be noted that due to dirt's opaque nature, root systems must be excavated to analyze them, limiting many root studies. This leads to my project, which involves studying the root architectural interactome, being the root-to-root interactions displayed by the physical space the root takes up. We'll go about this by capturing images of the excavated roots, running them through a digital pipeline, including softwares such as DIRT3D and Elastic, then creating a 3D image that we'll use for data analysis. As you can see, the images came out very similar to the original root, but to make sure that this system was accurate, my project was to take hand measurements to set a ground truth to compare between the computationally analyzed traits and my hand measurements. Some of the traits came out 1-to-1, but others not so much, indicating that more work was required on the system. Once the pipeline is finished, the system can be used in further studies, such as the one capturing root interactivity within the root architectural interactome, or in larger agricultural studies, such as automated systems capturing large data sets of root systems. The more we can learn about root systems, the better we can select for traits such as nutrient efficiency, allowing for us to reduce the fertilizer amount and thus its ecological cost. Thank you.
Hello everyone, my name is Anastasia Bradley, and this summer I worked in Dr. Zhu's lab where my project focused specifically on the STING protein, which is encoded within the STING1 gene. The STING protein is responsible for regulating the immune system's response to viral and bacterial infections. However, an overproduction of STING can lead to the growth and spread of tumors. So we wanted to see if we could create a plasmid tool where we could express the more easily detectable hybrid protein EGFP STING, which we would accomplish by combining the STING1 gene to another gene found in our target plasmid EGFP. The goal of this project is to garner basic information about STING and to better understand how it works so that researchers could use this for future studies regarding cell behavior in relation to STING levels. The first thing I did was use a program called pDraw, where I mapped out the genetic sequence of our insert, which is the STING1 gene, and our target plasmid, which we would be inserting the STING1 gene into, EGFPC3. We then ran PCR so we could generate multiple copies of our insert, and then running an R-Gross gel so we could verify the band-based pair lengths of our insert and our target plasmid. Afterwards, we cut the insert from the gel and ran it under a high-pressure centrifuge so we could extract the DNA solution from the gel and then cleaned up that DNA before combining it with our target plasmid into bacterial cells to multiply, and then later putting that same final plasmid into mammalian cells. We then use that plasmid to be multiplied in bacterial cells before finally putting the same final plasmid into mammalian cells. Overall, we were able to successfully implement the STING1 gene into our mammalian cells, into our final plasmid, and to observe the final protein, EGFP STING. We hope this can facilitate long-term drug development in the future and other studies. Thank you.
Hello, I am Bobby Briones, and I worked in Dr. Jane's lab over the summer to study redox molecules to be used in organics redox flow batteries. These batteries work under a chemical reaction known as a redox reaction, where chemicals undergo reduction and oxidation, or holding and accepting electrons to charge and discharge current. These batteries are superior for large-scale energy as they are economically and environmentally sustainable and long-lasting. We tested 5 chemicals and have found one chemical known as tris-cation as a potential candidate for a redox flow battery. We used 2 techniques known as cyclic photometry and differential pulse photometry. Cyclic photometry helped us understand the behaviors of the molecule, where we are able to understand the reversibility reaction and if the reaction can happen at all. The second technique, known as differential pulse photometry, is used to determine the operating voltage of the battery. For our experimental battery test, we use an H cell and use water as our solvent. We found that the coulombic efficiency meets that of lithium-ion batteries, but galvanic and voltaic efficiencies are lower due to the oxidation of water, reducing the output of the energy we put in. Overall, we have found that tris-cation, although it does work in water, still needs further research to be fully compatible with water. Thank you.
Hi, my name is Matt Capaldi, and this summer I worked in Nikolai Gulubev's quantum physics lab. Imagine you have a diatomic molecule and you can stretch and compress its bonds. When you do this, potential energy is stored in the molecule, as shown by this red line here and approximated by the black parabola. In this process, the nuclear geometry of the molecule is indefinite. However, a particle's position probability can be modeled using a wave function. These wave functions are completely analytically solvable using the Schrödinger equation. These wave functions also show likely transitions. Simply put, the similarity between 2 different wave functions and 2 different potentials show their likelihood of transition. I developed Python code to model 2 arbitrary bound potentials, the blue curve with its ground state energy level wave function, and the red curve with its first 10 energy level wave functions. The likelihood for the ground state wave function to transition to any of the 10 other wave functions is modeled by an FCF spectrum. In this example FCF spectrum, the most likely transition is to the 5th energy level, which would require 23 electron volts to make the jump. This can also be thought about in 2 dimensions, when manipulating the molecule in the x and y directions. I developed code to model this too. I then began investigating a real-life application of all of this by investigating the molecule methyl iodide transitioning from its neutral to its cationic state. Using various software, I was able to obtain this transition's FCF spectrum. The application of all of this work is understanding energy and particles on the atomic level, such as in controlling chemical reactions, which will have huge implications in the pharmaceutical industry. Thank you.
Hello, my name is Aiden Crabtree, and I worked in Dr. Maya Kalber's gut brain lab. We focused on understanding how the brain and the gut communicate via the vagus nerve. The vagus nerve is the neural pathway between the brain and the gut, where they communicate by using neuropod cells. A neuropod cell is a specialized cell found in both the colon and small intestine. Neuropod cells are used for the sensory of nutrition. This is why we were able to depict between artificial sweeteners and sugar and why we prefer sugar. My specific project focused on understanding how the brain communicated to these neuropod cells. The methods I used were fluorescence in situ hybridization, or FISH, to look at RNA sequences, and immunohistochemistry, or IHC, to look at specific pre- and postsynaptic proteins. We would then use a fluorescence microscope to look for green and red fluorescent light in our tissue samples. The results we found were that for FISH in the left column, we were able to see red fluorescence indicating where the neuropod cells were located, validating this method. In the middle column, we added antibodies to our presynaptic proteins, where we should have seen green fluorescence, which we did not. And in the right column, we added antibodies to our postsynaptic proteins, where we should have seen red, which we also did not. The reason IHC could not have worked is due to incorrect dilution percentages, which would have to be changed in future experiments. The reason we are doing this research is to understand how the brain communicates to these neuropod cells. This would be the first step to understanding how we feel affects how we perceive food. Thank you.
Hi, my name is Allison DeVaughn, and I worked in Dr. Capaldi's lab this summer. The TORC1 complex controls cell growth and metabolism, as well as nutrient transport and synthesis. I looked at proteins that may be downstream of TORC1 and visualized them using microscopy. I did this by adding a green fluorescent protein, or GFP tag, onto each protein. This allowed me to visually see the changes in abundance or localization made by each protein over the time period. I quantified these results. The main protein I looked at was PUT4, which is a proline transporter. I observed the change from glutamine, a high-quality nitrogen source, to 4 different conditions. In proline, a low-quality nitrogen source, and in minus nitrogen, a source with no nitrogen. Early on, the abundances in each cell matched, and this makes sense because the cells are trying to adapt to the new nutrient source. However, in proline, the abundance was able to level off at the later time points because the cells were able to adapt to the new nitrogen source, but in the source with no nitrogen, the cells were unable to adapt to the lack of nitrogen, and so the abundance kept increasing. In rapamycin, a potent TORC1 inhibitor, we would expect there to be some change in abundance over the time points if PUT4 was downstream of TORC1. But as there is no change in abundance over the time points, this leads us to believe that PUT4 is not downstream of TORC1. In minus glucose, a source with no glucose, it makes sense that the abundance remains relatively constant over the time points because glucose concentration mostly affects the pKa complex and not the TORC1 complex. Overall, understanding more about TORC1 signaling in different conditions can help us identify how mutations in TORC1 can lead to different diseases. Thank you.
Hi, my name is Avery Eliscu, and this summer I interned in Dr. Ganesh's lab, where we focused on optimizing biomaterials for degradable drug delivery scaffolds. A scaffold is something that holds a drug and delivers it to a specific part of the body. We're looking at tumors and releases the drug slowly over a period of time as the scaffold degrades, which would theoretically remove the tumor. To make these scaffolds, we created a bunch of different materials, and we found that the best material used was PEGDA 700. To then print those, we used a 3D printer and cured them in UV light. After curing them in UV, we put them in different conditions like temperature and pH, and the temperatures we did were 23 degrees Celsius, 37 degrees Celsius, and 42 degrees Celsius. All of these temperatures show different results, where the highest temperature had the fastest degradation and the lowest temperature had the slowest. 37 degrees Celsius was the optimal temperature for scaffold degradation. To check these results, we then looked at them in a nanodrop and under a microscope, and the FITC in the solution, which is a coloring agent, would show, and the more degradation, the more FITC has been released from the scaffold. These graphs show that at higher temperatures, the scaffold degrades fastest, and at lower temperatures, it degrades the slowest. You can see at 42 degrees Celsius, it degraded 100%, and the graph had a very high peak, whereas compared to 37 degrees and 23 degrees, it was very low and gradual. 23 degrees Celsius was a little too slow and did not release as fast as we wanted to, so 37 was optimal in the ranges between. The pH that we tested was most optimal in the acidic because it is lactic acid, and there is lots of lactic acid on tumors already. So it is very optimal to have 37 degrees Celsius and lactic acid pH for the scaffold. Lactic acid and a temperature of 37 degrees Celsius is the most optimal conditions for a scaffold to degrade at a gradual rate, and therefore is the optimal conditions. Thank you.
Hi, my name is Avani, and I worked in Dr. Kai's lab of neuroscience this summer. We focus on a part of the brain called the central amygdala and its connection to the circuit of eating. My project in specific looked at a condition called Prader-Willi syndrome, or PWS, a condition where affected individuals never feel full. Additionally, they also have low metabolism, making the weight they gain very hard to lose. PWS is a developmental syndrome where the symptom of uncontrollable eating developing around puberty. To look at this syndrome, we looked at a naturally occurring gut hormone called cholecystokinin, or CCK, that travels by the vagus nerve to the central amygdala, where it targets neurons called protein kinase C delta neurons, or PKC delta neurons. CCK activates PKC delta neurons, and when these neurons are activated, eating behaviors are typically turned off. Our aim in this project is to look at whether reduced activity of CCK and PKC delta neurons of a Prader-Willi syndrome model is what causes uncontrollable eating. To do this, the mice we were testing underwent a 24-hour fasting period, after which they were given either a saline control or a CCK treatment. Then, they were exposed to food for 90 minutes, during which we measured their food intake. After that, we processed the brain tissue using immunofluorescence, which is an imaging technique that allows us to view the activity of PKC delta neurons in a microscope, as shown in the figure at the very bottom. Our results showed that food intake in response to CCK decreased in both genotypes, our control mice and our Prader-Willi syndrome model. And as we can see on the left, where yellow dots indicate activity of PKC delta neurons, there was increased activity in response to CCK in both genotypes as well. Our overall finding was that there was a reduced effect of CCK in the Prader-Willi syndrome model. In the future, we would like to use calcium imaging techniques to further study these PKC delta neurons in real time, which would help us in understanding whether the satiation circuit is impaired or whether it's just these PKC delta neurons. Thank you.
Hi, my name is Sophie Gao, and this summer I worked in Dr. Karn's pharmacogenomics lab, where I studied the relationship between the gene PSG1 and warfarin dosing. Warfarin is a commonly used blood thinner where the dosage varies a lot per person based on factors such as diet, lifestyle, and genetics. Getting an inaccurate dose of warfarin could lead to harmful side effects such as excessive bleeding, so it's important that we find a way to predict a person's exact warfarin dose requirement. In particular, I studied the PSG1 gene's copy number variations, which are large replications or deletions of a certain gene. We chose to study PSG1 in particular because previous studies have shown that it might have a significant role in warfarin dosing. I studied around 100 DNA samples taken from warfarin-treated patients, and to determine the copy numbers of each of those, we started by quantifying them with a qubit fluorometer before I used qPCR to amplify the DNA and a Tachman assay to detect the specific gene sequence that we wanted to look at. Then we imported all of our data into CopyCaller, which is a software that determines the copy numbers and then generates a bar graph similar to the one on the left. As you can see, the majority of my samples had a copy number of 2, which is standard, but this does mean that I did not have a lot of variation within this particular cohort. Overall, I did not find a significant relationship between PSG1 and warfarin, but it is interesting to note that the samples that had a total deletion of the gene did have lower warfarin dosages than the other ones, which does align with previous studies. However, since there were so little samples that had a total deletion, we were not able to reach a definitive conclusion about this. In the future, if we were to continue experimenting with PSG1 and warfarin, we would probably want to test a larger amount of samples just so we could have more variation within the gene. Thank you.
Hi, my name is Sarah Goldman, and this summer I was working in Dr. Pierce's lab. In his lab, I was analyzing astrocytes in the brains of mice with Alzheimer's disease compared to a healthy mouse brain. Astrocytes are cells in the brain that contribute to many different functions, but importantly, they play a key role in inflammation and maintaining the blood-brain barrier, which is a barrier that keeps harmful substances out of the brain and beneficial ones in. But in a brain with Alzheimer's disease, astrocytes are harmful to the brain and contribute to the disease's progression. To analyze the astrocytes, I used immunohistochemistry as my method. Immunohistochemistry is a method where you use antibodies to tag a specific protein in the cell, and the antibodies are linked to a fluorescence dye so that the proteins can be imaged through a fluorescence microscope. The antibodies that I used were looking for GFAP because that is a protein that's very abundant in astrocytes. When using the immunohistochemistry, I was able to gather images from a brain of a mouse with Alzheimer's disease and a healthy mouse brain in the cortex and hippocampus parts of the brain. Then we used ImageJ to calculate the percent GFAP coverage, which is the surface area that is covered by astrocytes in the brain, which is important because that shows astrocyte reactivity. In the cortex, we found that the percent GFAP coverage was higher in the Alzheimer's mouse model than the wild type, and in the hippocampus, the wild type had a higher percent GFAP coverage than the Alzheimer's mouse model. To further analyze astrocytes, we can look at their skeletons and look at the branching, because astrocytes branch more when they're inflamed, which could give us a better idea of their reactivity. Thank you.
Hi, my name is Ethan Hancey. Over the summer, I worked in the soil lab under my mentor, Helena Harbin. In our work, we hope to improve pancreatic tumor margin identification. Pancreatic tumors have a low 5-year survival rate and are difficult to preserve healthy tissue during surgery. So through our work, we hope to improve patient and surgical outcomes. In Figure 1, we injected a human pancreatic tumor cell line into a mouse. Cultivating and sampling the mice, we then imaged it under an imaging modality for certain chemicals such as FAD, which is related to cellular metabolism. An example is Figure 3, which is an example image of FAD. We then used a Python graphic user interface to select regions of interest of tumor, margin, and normal. Then using these regions of interest, we conducted Herlich features, which are a set of mathematical methods that analyze the pixel values of the regions of interest and set them into distinctive values. We hypothesized we can use these Herlich values to find correlations between the tumor, the margin, and the normal regions of interest. Shown in Figure 5, the heat map visualizes the statistical significance of the Herlich values computed. FAD was shown to be one of the best channels for imaging and margin identification. Specifically, Herlich features 3, 6, 8, and 12 were shown to be the most statistically significant in margin identification, which we then used the values of these Herlich features to then train a machine learning model, which showed promise. We hope in our future work to further fine-tune this machine learning model and translate this work over to more aggressive forms of pancreatic cancer to improve surgical and patient outcomes. Thank you.
My name is Alexis, and this summer I had the opportunity to work in Dr. Kim's red blood cell lab, also referred to as the RBC lab. In this lab, we investigated different underlying principles of microparticles and proteins that could be suitable for the cytoskeleton of a red blood cell. Currently, there is a growing need for RBCs, primarily because of donor shortages and consistent struggles with transporting and storing blood. Globally, transfusions are a requirement for surgeries, car accidents, cancer treatments, and more. However, donated blood is often insufficient to meet this demand. That's why artificial red blood cells are a promising alternative that offer a long-lasting, stable, and are universally compatible solution. In my lab, I specifically worked on expressing and producing 2 types of proteins, spike-tag proteins, which are binding proteins, and spike-catcher proteins, which are long, flexible proteins. It was important to find a balance of the 2. The more rigid proteins help the RBC maintain structure and function, and it was also important to note that the more flexible proteins allowed them to travel through blood vessels. We did this by inserting a target DNA sequence into E. coli, as it can live in humans. We did this while optimizing temperatures at 30 and 37 degrees Celsius, and we also managed expression rates for IPTG amounts. We noted that the more stiff proteins produce best at 37 degrees Celsius, whereas the more flexible proteins produce best at 30 degrees Celsius. We also then combined the proteins that were optimized using spike-tag and spike-catcher ratios. We did this to see whether the proteins would form long chains suitable for the cytoskeleton of a red blood cell. We found that the higher order oligomers performed best. In conclusion, it was important to note that it was a balance of rigidity and flexibility that helped to create the cytoskeleton of the red blood cell. In conclusion, this research will show that fine-tuning parameters such as incubation temperature and expression rates will produce the optimal proteins that can emulate red blood cells. Thank you.
Hello. My name is Abigail Hernandez, and this summer I was an intern in the lab of Dr. Vinesh Subbion at the University of Arizona's College of Engineering. My lab's focus is on how human and machine interactions can work together to improve safety outcomes in the medical field. My project specifically focused on how human and safety interactions, such as young drivers and safety automation features, can work together to improve safety outcomes in fatal crash risks. In Arizona alone, 1 in 6 crashes are caused by young drivers, 16 to 24, who have had their license for less than 5 years, even though they only account for about 11% of drivers. 34% of crashes that are fatal, that are caused by this age group, are single vehicle, meaning that they are not accidents. They are preventable, usually caused by loss of control or distraction. Utilizing data, we were able to develop an app called Driver Mentor Mode, a device feature that would allow young drivers to get real-time proactive feedback. Most modern safety features in cars are reactive, meaning they only give feedback after the incident has occurred. Proactive feedback would mean drivers would be able to assess the situation before it happens and before that 2 to 3 second decision-making window when most crashes occur. By using this app and implementing it into better drivers' education programs, Arizona would not only become a leader in driver safety, but would save 1,430 families from the devastating phone call that they would never want to receive. Thank you.
Hello, my name is Gracie Im-Pyun. This summer I worked in Dr. Lee's lab focusing on protein inhibition for drug discovery. Purine biosynthesis is a pathway that creates purines, components of DNA and RNA, and energy like ATP. Because of this pathway's active nature, its presence in bacteria and pathogens can lead to their proliferation and survival. PureB is an enzyme present in this pathway and has been linked to the bacteria Mycobacterium tuberculosis, MTB, which causes tuberculosis, a worldwide infectious disease. My project tested 3 bisphenol compounds to test their inhibition activity against PureB. Previous research has shown that bisphenol is an inhibitor of PureB, thus why we are testing variations of it. Firstly, we performed a bioluminescence assay measuring the enzymatic activity of 3 enzymes, PureB, PBDK, and luciferase, to test whether our analogs are a true inhibitor of PureB, in which case the analogs would only inhibit PureB and not PBDK or luciferase. And this was measured through the luminescence of the reaction produced. Next, we did a thermal shift assay, which measured the binding stability of the analogs plus PureB. This was measured through fluorescence. Lastly, we performed a cytotoxicity and antibacterial assay, which measured the toxicity of the analogs with cells, and an antibacterial assay, which measured their effectiveness against the bacteria MTB itself. Our results showed that all 3 analogs showed inhibition against PureB, and specifically, analog 2 had the highest inhibition. In the top left corner, analog 2 has the lowest IC50, which indicates that it has the strongest inhibition because it requires the least amount of concentration to inhibit PureB's activity by half. These results can be used in future experiments to further test PureB's activity as a potential drug target for tuberculosis. Thank you.
Hello, my name is William Jacobsen, and this summer I worked in the Frost Lab, which focuses on integrative and applied chemical ecology. My project looked at if plants emit volatile compounds in response to herbivory, and if other unwounded plants can detect and respond to those compounds. I worked with a specific class of volatile compound called greenleaf volatiles, or GLVs, which comprise the smell of freshly mown grass. GLVs are emitted by plants in response to herbivory or damage. First, in order to gain a better understanding of how plants respond to herbivory, we sampled VOCs emitted by wounded and unwounded plants in Figure 2. Then, for our second experiment, we treated plants with either a control or a GLV vial. Then, we allowed caterpillars to feed on one half of the plants for 24 hours and then quantified the plant's phytohormones using gas chromatography mass spectrometry. For our first experiment, we found that wounded plants emit a diverse set of volatile compounds that unwounded plants do not emit. Among these include greenleaf volatiles and other complex herbivore-induced compounds. Additionally, we measured volatile emissions from wounded and unwounded plants in 2-hour intervals across 2 days, and we found that wounded plants increase volatile emissions over time. For our second experiment, we first assured that all plants received consistent damage. We found in our second experiment that plants previously treated with the GLV signal enhanced jasmonic acid production in response to herbivory. However, the GLV signal did not directly induce jasmonic acid production. This indicates that the GLV signal primed the plants for future herbivory. This work deepens our understanding of plant resilience, communication, and innate immunity in agriculture. Thank you.
Hi. My name is James Jirousek, and this summer I've had the opportunity to study in Dr. Hunter's lab under the mentorship of Edwin Umanzor. I've been studying the leaf-footed bug and 2 different bacterial symbionts that impact the bug's development. The two bacterial symbionts in question are Capaldi, a nutritional bacteria that is housed in the leaf-footed bug's midgut, which is vital for it to progress into adulthood, as well as Lariscella, an intracellular bacteria that is passed down from mother to offspring. The questions I've been trying to answer this summer are, how do different strains of Capaldi affect development and reproduction, as well as can Lariscella assist a suboptimal strain of Capaldi in development? We tested this by having 6 different sets of the leaf-footed bug, one with LZ01, one with LZ24, one with LZ28, 2 with LEP1P3, a suboptimal strand, one had Lariscella and one did not, as well as one control group. We found that LZ01, LZ28, and LZ24 were very beneficial for the bug. They were able to develop into adulthood in less time and weighed more. However, regardless of Lariscella status, LEP1P3 continued to perform suboptimally. However, it is important to note that because of the time restraints on our project, we weren't able to collect all of the data for LEP1P3, only about 20%. In the future, we would continue this experiment by waiting for all of the bugs to reach adulthood before collecting data, as well as quantifying the amount of Capaldi in the midgut by using qPCR to assess whether there's correlation between amount of Cab and the weight and / development of the bug. Thank you.
Hello, my name is Ashley Zhou, and I worked in the Gardner Chen lab under Dr. Kellen Chen, where we primarily investigated how diabetes affects wound healing. So over 200 million of the 830 million people with diabetes have impaired wound healing. This means that diabetic cells can't respond to insulin, which leads to high blood sugar levels and a weakened immune system, which weakens wound healing in general. However, some individuals are able to heal effectively, and we wanted to see why. Our past research on mice showed that there was a high expression of these immune-related proteins CD14 and CD64 in the non-diabetic tissue, and a low expression of those in the diabetic tissue. Therefore, my lab specifically applied this to humans to see if the trend was similar. To do this, we first collected tissue samples from non-diabetic, which is the control, the diabetic healing, and the diabetic non-healing from consented patients. After that, we froze the samples, and then we sectioned them by using a cryostat, and then stained them, and then we imaged them using a confocal microscope that allowed high resolution for us to see the proteins clearly. Then we quantified them by using a MATLAB code developed by our lab. And the results show that there was actually a higher expression of CD14 and CD64 in the control tissue, and the difference was really significant in contrast to the diabetic groups. However, something unexpected we found was that while there was a higher expression of CD14 and CD64 in the diabetic healing in contrast to the non-healing, the difference was not significant. For future reference, we plan on seeing how these proteins interact in the wound microenvironment so that we can use them for future treatment. Wound healing is a natural process that is impaired in diabetic tissue. However, through our research, we're closer to finding the right treatment so that these individuals can have the same opportunities of healing fully. Thank you.
Hello, my name is Tyler Johnston, and over the summer I worked in Dr. Zhu's laboratory where I created a plasmid vector tool, or a double-stranded DNA molecule that can be used to study tau protein in mammalian cell cultures. Tau is a critical neuronal protein involved in stabilizing the microtubules, or vertical filaments that make up a cell's outer skeleton. However, in some people, tau can form insoluble aggregates that can result in a classification of neurodegenerative diseases called tauopathies. Most notably of these include Alzheimer's and Pick's disease. I amplified a pre-existing plasmid containing my tau insert and my target vector that I would insert the tau protein into. I then performed a restriction digestion using ECO-R1 and BAMH1 cut sites that would allow me to stick both the insert and the final plasmid together in a process known as ligation. To confirm a successful cloning, I first ran my insert, or PCR product, on an electrophoresis gel to confirm if the insert was the estimated length of 1.3 kilobase pairs. After confirming that and going along with the restriction digestion and getting my final product, I ran my final product on a similar electrophoresis gel, as shown in Figure 6, through the cloned 3XT clone 3 and 3XT clone 5, my best performing clones. As you can see, both these clones had a resulting length of 7.3 kilobase pairs, which matched my expected one in figure 4. These electrophoresis gel results show both that the insert was in its estimated proper length and that the final product matches the expected length. Finally, I sent my final product off to sequencing, where I compared my expected results to the returned results, which were a success. My final product can then be used in mammalian cell cultures to study how tau reacts under normal baseline conditions and stressful conditions that would hopefully lead to better therapeutic treatment development and a greater understanding of the tau protein and tauopathies. Thank you.
Hi, my name is Anika, and this summer I worked in Dr. Gutschev's Biosymbiotic Sensors Lab. This lab focuses on creating wireless, battery-free, implantable devices for clinical-grade diagnosis. Previous research has shown us how our skin reacts to different stressors, like exercise, by studying the skin gases and moisture emitting from the blood vessels. Gases like CO2, volatile organic compounds, and water vapor are being collected in a leaky cavity that is placed on top of the skin, where they're then analyzed by the sensor and compared to ambient conditions. The schematic on the right further illustrates this principle by showing how ambient air is diffused through the leaky cavity, preventing any concentration buildup. This research could potentially help us find metabolic and health conditions faster. This principle was effective on on-body testings. A stationary cycling test was conducted where the sensor was placed on one arm and a cotton swab was placed on the other arm. The cotton swab was to be weighed and replaced every 2 minutes to be served as our gold standard and compared to our sensor readings. This test was run under moderate conditions using a heart rate sensor as our indicator. The results given by the sensor and cotton swab were pretty similar, giving us accurate results showing that the sensor did work. But one thing we found out was that when humidity was too high, the sensor would tend to give inaccurate results. That's what I'm trying to fix. My goal is to try to create a membrane, which is the box the sensor is in, that would be able to accurately find data in any type of condition, no matter how humid or dry it is. Thank you.
Hi, my name is Srihan Kondle, and this summer I interned in the lab of Dr. Ross Buchan, which studies the clearance of stress granules. Stress granules are non-membrane bound organelles which form in cells in response to various stressors. G3BP1 and G3BP2 are two RNA membrane proteins which assemble stress granules. G3BP1 and G3BP2 have also been linked to cell cycle regulation by means of regulation of P53 and P52 break proteins. Previous Buchan lab studies have shown that G3BP mutants affect cell cycle re-entry following S-phase arrest. My project focused on how G3P mutants affect the cell cycle re-entry following G2/mitosis phase arrest. To do this, we conducted 3 tests, the first of which was flow cytometry to measure propidium iodide fluorescence and scatter to measure DNA content and cell cycle phase. We also ran a cell death assay to measure the survival after G2M phase release. Finally, we ran a Western blot to assess CDK activity at the G2M phase using CDK2 and CDK substrates. The results show that in the R0 release cells, the G3BP2 single mutant showed the greatest change in PI positive cells, and there was no noticeable change in the other cell lines. The Western blot data shows that there is an indistinguishable CDK band between G3BP1 single mutant and wild type, but there was a noticeable change in G3BP2 and G3BP1 and 2 double mutants. This research is important because understanding G3BP1 and G3BP2 can help determine drug targets to treat various cancers. Future experiments can include analyzing these cell lines with other cell cycle inhibitors, including CDC25C, CDK1, and cyclins, in these same cell lines to discern possible effects underlying G3BP mutant effects on cell cycle progression. Thank you.
Hi, my name is Keertana Konkimalla, and this summer I had the opportunity to research in the Thacker lab, where we studied the role of 2 novel mutations in the ADCK4 gene. The first mutation, I346S, is when the amino acid isoleucine is mutated into the amino acid serine. And the second mutation is W520X, when the amino acid tryptophan is mutated into a stop codon, therefore truncating the protein. ADCK4 plays a key role in the biosynthesis of CoQ10, an important mitochondrial molecule necessary for ATP production, especially in high energy demanding organs such as the kidney. Mutations in this gene often result in severe mitochondrial dysfunction and other serious health conditions. In fact, a patient recently developed steroid-resistant nephrotic syndrome and eventually end-stage renal disease due to inheriting both of these mutations. In order to test this, we grew human podocyte cells, which are specialized kidney cells, and transfected mutated DNA in order to measure the amount of CoQ10 in each of these cells. Preliminary data shows that both mutations alone show negligible amounts of CoQ10. However, surprisingly, when both mutations are present, there is a measurable amount of CoQ10. This signifies that these mutations have an effect on CoQ10 biosynthesis. By studying mutations such as these, we can better understand the structures and functions of the ADCK4 gene and its role in cellular processes. Thank you.
Hi, my name is Heather Lee, and over the summer I worked in Dr. Hutchinson's multi-scale brain imaging lab, which researches the validation of non-invasive biomarkers of pathology like Alzheimer's disease through MRI strategies. Alzheimer's disease is a neurodegenerative disease that affects memory and thinking. Patients are typically diagnosed after symptoms appear, which at that point they have already experienced severe brain damage. The disease is very difficult to diagnose at early stages due to very subtle brain changes prior to the presence of symptoms and the lack of a single definitive assessment. My project specifically aimed to verify MRI metrics with microscopy imaging focusing on beta amyloid plaques. These plaques are protein accumulations and biomarkers of Alzheimer's disease that are mainly found in the hippocampus, a region where damage is linked to the disease's symptoms. I imaged post-mortem temporal lobe specimens with a microscope at 20 times magnification, with our specific region of interest being the hippocampus subfield CA1. After microscopy imaging, I used an image analysis software to collect data about plaque count and plaque area. I then used this data to compare with MRI metric data in a correlation analysis. We found that the plaque count and MRI metric correlation was stronger than the plaque area and MRI metric correlation. Additionally, we found the strongest correlation between plaque count and BPF and plaque count and T1. This suggested that the relaxometry metrics, which were T1 and BPF, had higher specificity for the plaques than the diffusivity metrics, which were FA and MSD. While more research is needed in other hippocampus subfields and other brain regions affected by Alzheimer's disease, like the entorhinal cortex, validating biomarkers with MRI strategies can further develop early diagnostic methods of the disease. Thank you.
Hi, my name is Arianna Malik, and this summer I did research in Dr. Pier's lab where we studied the role of vascular function on cerebrovascular disease. Specifically, we looked at the role of the expression of ApoE4 on the reduced contractile ability of cerebral arteries in mice. Cerebral vascular disease, or CVD, are conditions that impact blood vessel function and thus cerebral blood flow regulation. Our preliminary data show that ApoE4 has been known to lead to a reduced contractile ability of cerebral arteries in mice. The contractile ability of these cerebral arteries are dependent upon L-type voltage-gated ion channels, or CAV 1.2 channels, in vascular smooth muscle cells. However, whether the number or the function of these channels becomes impaired in the presence of ApoE4 remains unknown. In order to test our hypothesis that the presence of ApoE4 leads to reduced expression of CAV 1.2 channels, we used immunohistochemistry to locate these channels and quantified them using image Fiji. Upon initial investigation, we found there to be a significant difference in the number of channels present in the ApoE4 versus the ApoE3 cells. In the ApoE4, there were significantly less channels compared to the ApoE3. However, when we compared these to the cell size, we found there to be no significant difference in the number of channels present, as the number of channels was changing due to the cell size rather than the expressing gene. In the future, obtaining a larger sample size and higher resolution images will aid in forming a definite conclusion. Furthermore, studying the activity of these channels alone will aid in CVD research, as ApoE4 remains a risk for vascular dysfunction and CVD can lead to higher cognitive impairment. Thank you.
Hi, my name is Melayna Matheson, and this summer I worked in Dr. McGee's biomedical engineering lab in the Cancer Center. I made a perfusible brain on a chip using granular hydrogels. My brain model improves on current brain models by incorporating flow and structure. The most common current brain model is an organoid, a spherical structure of densely packed cells. The model has a few limitations. The cells are so densely packed that it's unlike anything you would find in the body. And the cells in the center of the organoid die because they aren't receiving enough oxygen or nutrients. My model addresses these problems by using granular hydrogels, tiny solids made mostly of water. The granular hydrogels allow my model to stay in 3D instead of 2D, so the cells can continue to act similar to how they would inside the body. Granular hydrogels also allow flow of nutrients throughout the model and flow of oxygen so that cells are not dying. I used PC12 cells. PC12 cells are cancer cells that, when given nerve growth factor, will start to look like neurons. The long outgrowths that you can see on my slides that they grow are similar to dendrites and axons that neurons have. Because the PC12 cells can start to look like neurons, we can use them in the brain model. I mixed PC12 cells with granular hydrogels and plated them in a fluidic chip. Then I ran nutrients through the chip and included nerve growth factor so that the cells could start to change. Over the next 8 days, I imaged the cells as I grew those neuronal outgrowths. Now that we have a model of healthy tissue, in the future we are going to add cancer to this model in order to study the effects it has on healthy tissue and make patient-specific treatments. Thank you.
Hi, my name is Finnegan McGill, and this summer I worked in Dr. Pascal Charet's lab looking at cell migration. Cell migration is an important aspect in many cell behaviors, from immunology to cancer cell metastasis. That's when cancer cells move away from their original cancer site, making them more deadly and more dangerous. Now, what causes those cancer cells to move? Well, that would be the cell migration pathway, and what happens is that a cancer cell gets a chemical signal, a chemoattractant, that it recepts on the cell membrane. Then a complex of proteins called mTORC2 localizes at that cell membrane and then phosphorylates or activates a whole bunch of different proteins that are connected to cell migration. There are 3 different proteins that I looked at: AKT, PKC delta, and PKC zeta. The PKC family of proteins has been linked to cancer cell aggression and movement. And what I did for my experiment is that I looked at 3 different chemoattractants and stimulated my cells at different time points to create a behavior curve for the activation of those 3 proteins. And what I found was that each chemoattractant stimulated the cell migration pathway differently, giving different peaks in peak phosphorylation and different trends in that behavior curve. This allows us to get a better understanding of the entire cell migration pathway as a whole and potentially develop a drug or a targeted strategy to prevent cancer cell metastasis. Thank you.
Hi, my name is Cambria McLeod, and this summer I had the opportunity to work in Dr. Taylor's lab, part of which is focusing on the creation of glide traps. My specific project focused on the synthesis of the ingredients to make these glide traps, which are called 3-mercapto 1H indoles and have 4 and 6 carbonitriles substituted in them. The 4 and 6 indicate the place on the molecule where the carbonitrile is substituted in. These glide traps are used to study protein-protein interactions on a small and specific scale. When we created the molecules for our reactions, they were trapped in an aqueous layer, which is not useful to the lab. So in order to separate them out, we put them in a separating funnel with organic solvents in order to extract the molecule from the solution into a form we could use. We then evaporated out all the solvents and ran the remaining solid through a column chromatography, which separates molecules by polarity. After that, we were able to extract the molecules we wanted with water by precipitation and then filter it using a filtration flask and vacuum, so that all we were left with was the molecules we wanted. Moving on to the results, we got results in the form of NMR analysis, which looks at how protons are arranged around a nucleus to help identify a molecule. There are 2 kinds of NMR, proton and 13C analysis. For the 4 substituted molecule, we were able to run both forms of NMR, and both confirmed the presence of the molecule we were looking for. For the 6th substituted molecule, we were only able to run a proton analysis, which did confirm the presence of our molecule, but with some impurities, so the reaction went wrong at some point. The implications of this in future research are that now those mercaptoindoles can be used to synthesize glide traps in the lab, and those can be used to study specific protein interactions, which can have impacts in drug delivery and other medicine, as well as food engineering. Thank you.
My name is Fabian Melton, and over the summer I had the opportunity to work in the Reilly lab under my mentor, Brendan Risk, and we studied a genetically modified variant of the Anopheles mosquito to attempt to find a both ecologically friendly and foolproof way of inhibiting the spread of malaria. Because the malaria parasite develops within the midgut of the mosquito using an essential nutrient known as vitamin B5, which itself can only be acquired from the mosquito, we inserted an extra PANK-K gene inside of the mosquito, which would deplete the supply of vitamin B5 available within the midgut, starving the parasite and killing it before it's able to spread. We had two essential questions about this gene: whether or not this gene affected the fitness of the mosquito, and where and when this gene is being expressed. To analyze this gene's effect on fitness, we looked at both the average amount of eggs laid by both the wild-type population and genetically modified population, and also looking at the percentage amount that did and did not lay eggs for both populations respectively. To figure out where and when this gene was being expressed, we used Western blotting to figure out when, and PCR for cDNA analysis to figure out where, using samples of both genetically modified and wild-type midguts and carcasses, and for Western blotting, after a blood meal. Our data concludes that this gene has little to no effect on the amount of eggs laid by the mosquitoes, and is being expressed in the proper place at the right time. This suggests that this method of control for malaria is worthy of further investment. Thank you.
Hi, my name is Brian Wing, and this summer I worked in Dr. Laura Meredith's lab. Her lab specializes on how microbes affect the chemistry in our atmosphere. In particular, my lab focuses on hydrogen gas, and that's what the focus of my project was on. Hydrogen gas is considered an indirect greenhouse gas, meaning that it helps direct greenhouse gases such as ozone and methane to persist longer in our atmosphere. In my project, we wanted to see whether or not soil microbes, who already consume 80% of global hydrogen, could consume more with biostimulation. Biostimulation is the process of adding external soil to regular soil. This external soil has already been previously exposed to hydrogen, and we're hoping that the microbes in the external soil can grow and help the regular soil increase their consumption of hydrogen. So what did we find? Well, we found that biostimulation does work in very specific circumstances. As you can see from the top graph, elevated hydrogen, which means external soil already pre-exposed to elevated amounts of hydrogen, was very effective in increasing hydrogen consumption in regular soil. However, atmospheric and control soils weren't as effective. Of course, the site, can change how much hydrogen is consumed, but overall, the general trends still apply. Now, knowing this, what can we do? Well, we can conclude that the biostimulation is an effective tactic, and this could be used in potential engineering solutions such as a biofilter in mitigating excess hydrogen consumption and mitigating the risk of climate change. Thank you.
Hi, I'm Titus Nguyen, and I've been studying the acousto-optic effect in the Dr. Elkavash Optics Lab. Essentially, when a membrane has a sound wave blasted through it and you shine a laser through it, that laser will bend. And essentially, since pressure will then change the speed of sound in the membrane due to the fact that pressure will change the density of the membrane, you can relate the diffracted angle of light to pressure. And so by using this application, we're able to measure pressure in something like the retina. Currently, ophthalmologists believe that high intraocular pressure in the retina is linked to macular/retinal degeneration. However, it is currently assumed that retinal and corneal intraocular pressure are the same. And of course, this warrants a new method of measurement. And so for my experiment, we used something called a PDMS membrane, which is a vinyl-like substance known for its transmissibility flexibility, and efficiency at propagating ultrasonic waves. And to propagate those waves, we use something called a piezoelectric transducer, which converts electricity to mechanical waves to make something called a SAW, a surface acoustic wave. And so, by knowing the properties of PDMS, such as its density and elasticity, we're able to derive an equation using known principles such as double-slit diffraction. Based on the known properties of PDMS, such as density and elasticity, we were able to derive an equation using known principles such as double-slit diffraction. This equation then informed us about our experiment, which was divided into 2 phases. For the first phase of our experiment, we would determine the efficacy of the acousto-optic effect to determine pressure by using stretch as an analog and then varying that stretch and then relating that to the diffracted angle. And if this were to fit the curve, then we would move on to phase 2 of our experiment, which was to keep a constant stretch, such as something like 10 centimeters, and then varying a different variable like laser wavelength. So unfortunately, due to shipping delays, we were unable to conduct the experiment, but I hope to keep working with my lab in the future. Thank you.
Hi, my name is Zina Nwafor, and this summer I worked with Dr. Fabian Hernandez on the Mind After Midnight study. Research has shown that suicides occur disproportionately at nighttime. My lab is trying to figure out what exactly happens in the brain at night that makes people more likely to have thoughts about harming themselves. After participants' eligibility for the study has been confirmed and informed consent has been given, their activity, sleep patterns, and feelings will be monitored at home and in lab for multiple weeks. On lab study nights, participants will receive a collection of questionnaires known as the neurobattery to assess their mood at different times of day. Based on previous literature, I coded surveys on REDCap in order for participants to be able to describe their feelings quantitatively. For the Karolinska Sleepiness Scale, I used a single field Likert scale for the participants to rate their sleepiness on a scale from 1 to 9. For the Beck Hopelessness Scale and the Beck Scale for Suicidal Ideation, I used multiple fields as each question had unique answer options. Specifically, for the Beck Hopelessness Scale, I utilized reverse coding as the statements were a mix of both positive and negative statements. For positive statements, a response of false had to be scored as 1. That way, higher scores are more indicative of hopelessness. Lastly, for the varieties of inner speech questionnaire, the interpersonal needs questionnaire, and the positive and negative affect schedule, I used a matrix of fields as each of the options were scored on the same Likert scale. These questionnaires are important for the participants to be able to describe qualitative emotions such as hopelessness and suicidality using quantitative numbers. That way, it can support the data analysis the researchers need in order to confirm the connection between being awake at night and suicidal thinking. This study's findings can help guide suicide prevention mechanisms and imply that we need more suicide prevention resources for participants and patients during the nighttime. Thank you.
Hello, my name is Maya Perez, and this summer I worked in Dr. Shively's lab where my project's focus was on creating nanostructures in 2D material. 2D materials are atomically thin sheets that lose a third spatial dimension and confine electron movement to a flat plane. This confinement can change the chemical properties of substances. For my project, I used graphene, which is one layer of carbon atoms. The goal of my project was to create a nanostructure consisting of extremely small holes, about 1/1000th the size of a meter in width, in graphene. To do this, we used focused ion beam milling. This technique involves shooting helium ions directly at a surface to cut through it. To prepare graphene, we use the tape method. This involves repeatedly peeling back layers of bulk graphene until thin layers remain. That material is then transferred onto chips and examined under a microscope. Once we find chips that look clean and thin, we use atomic force microscope to gain nanoscale resolution of its surface and height. Once graphene is found and then patterned, it will serve as a top gate in a 2D capacitor-like device designed to trap electrons. Its unique structure bends the out-of-plane electrical field in the incident layer below. It creates energy wells that attract and trap electrons to confined space. In essence, the goal was to create a nanoscale environment where electrons can be controlled with precision. But why is that important? This method enables a 2D capacitor-based single photon emitter, which can be revolutionary for the next generation of quantum computing and communication. This method also showcases the ability to manipulate particle behavior and quantum location. In summary, my project showcases how patterning certain structures in 2D materials can be revolutionary for the next generation of science. Thank you.
Hi, my name is Adrian, and over the summer I worked in Dr. Leroy's laboratory working with nanomaterials to specifically make devices. So currently, there's a societal push towards nanotechnology, or more specifically with nanoscale devices at its forefront, which simply put is stacks of atomically thin materials. One of the biggest problems with research in this field is the lack of a replicable and reliable method for the fabrication of these devices, which is where my research came into play, where my team and I fabricated a device using a replicated 4-step method. The device we created was a repeating structure, as shown on the left, of atomically thin graphene, which is hexagonal structure of carbon atoms, and bulk hexagonal boron nitride, which is the same structure, but it uses boron nitrogen instead of carbon. Throughout this entire process, we made sure to ensure quality through 3 main factors: that being a consistent uniformity across the piece, a maximized size of the piece or contact area, and then a minimized residue around the piece or defects. But how do we actually fabricate the device? In the first step of the 4-step process, separation, we removed an atomically thin layer from a bulk material using the Scotch tape method, and then we placed it on a silicon wafer. In the next step, the searching step, we looked under an optical microscope where the color of the piece would give insights into thickness. Then in the second to final step, we made sure to use an atomic force microscope where we're able to measure nanoscale variations in height across the piece in order to confirm its thickness and also its uniformity. Finally, we actually created the device in the end using the dry transfer technique, where we use a viscoelastic stamp and heated it up or cooled it down to make sure we were laying the pieces on each other as precisely and slowly as possible. In the end, we successfully fabricated a device as shown in the bottom of the slide. But why is this important? It's important because it provides a foundation for scalable and reliable device fabrication in the future, which is an essential step in next generation electronics, such as those in our cell phones in our pockets, as well as quantum measurements in the lab. Future research can work towards automating this process, making it faster, as well as work with more sensible materials like transition metal dichalcogenides. Thank you.
Hi, my name is Siyaa Poddar. This summer I worked with the IM-Pyun group researching elemental sulfur's role in optical materials. Elemental sulfur is the byproduct of fossil fuel refining, with very few industrial applications, despite 70 million tons being produced yearly. However, sulfur-sulfur bonds have high infrared transparency, particularly in the medium and long wave region, making them ideal for optical materials and thermal imaging. Additionally, sulfur itself is quite small and has a high refraction index, or ability to bend light. Therefore, integrating sulfur into polymeric optical materials, such as polysulfur NB2, allows the final product to be thinner, thus using less plastic and being more sustainable overall. However, one limitation of using elemental sulfur is the presence of tar impurities from the fossil fuel refining stage. These impurities can lead to light scattering, or the divergence of light waves, and increased optical absorption due to discoloration of the final material. Both of these phenomena in tandem lead to decreased optical transparency and lower performance overall. This study polymerizes sulfur of 3 differing purity degrees with the organic comonomer NBD2. The polysulfur NBD2 was cast into IR windows, and using Fourier transform infrared spectroscopy, we found that the sulfur with the highest degree of purity showed the greatest amount of transmittance across the spectra, particularly in the long wave region, which is ideal for thermal imaging. Additionally, using UV-vis spectroscopy, we only found the presence of light scattering within the inconsistent curvature of the low purity sulfur, with high purity sulfur showing no clear evidence of scattering. Thermogravitational analysis, which establishes the thermal degradation point of a material, and differential scanning kilometry, which establishes the glass transition states of the material, were used to ensure that polysulfur NBD2 was properly synthesized and that the organic comonomers were fully reacted. In sum, this study supports the use of high purity sulfur as a competitive feedstock for sustainable, low-cost optical materials. Thank you for your time.
Hello, I'm Sara Porter, and this summer I worked in Dr. Hunter's entomology lab with Leptoglossus zonatus, or the leaf-footed bug. This bug needs Capaldi, a symbiotic bacteria, in order to develop properly. This bug must get this bacteria from the environment every generation. Our lab aims to find where and how this bug acquires the bacteria, and if they use smell to do so. We used a Y-tube olfactometer that runs odors for the bug to move towards one. We ran 3 sources of the bacteria through this: frass, or bug feces, bug-exposed soil, which contains the frass, and clean soil, which has less of the bacteria. Our results were not what we expected, as the bugs chose the controls over the sources of frass, and the bugs seemed to be neutral towards the clean soil. We theorized that this may be because the bugs use a different sense than smell to find the bacteria, or that they are trying to avoid a gut parasite recently found in the lab. Thank you.
Hello, my name is Nick Rice and I worked in Dr. Sandhu's physics lab. We essentially studied the movement of electrons, which is important because it underlies every single chemical reaction that happens. If we understood this better, we might be able to control chemical reactions, which would lead to advancements in material and drug design. The problem with electrons is that they move extremely fast on the attosecond timescale. 1 attosecond is a billionth of a second. If you were to compare an attosecond to a second, it'd be like comparing a second to the entire age of the universe. In order to measure something this fast, we need to use ultra-short pulses of light generated by a high-powered laser. These lasers are so precise that small changes in atmospheric temperature and pressure cause the laser to drift, which interrupts experimentation and requires manual adjustment that wastes time. The goal of my project is to correct for laser drift by creating an automated position feedback loop using a camera mirror system in the programming language LabVIEW. First, I programmed the camera to automatically adjust the exposure so that the brightness or intensity was in a range that we can manipulate. Then I characterized the laser by creating a Gaussian for it, which is essentially a profile that tells us things like its size and amplitude. Then the camera finds and tracks the maximum intensity. If the max intensity is too far from the target, the camera tells the mirror to move the laser towards the target until it is within an acceptable range. The laser moves in the Y direction first, then the X direction, and slows down as it gets closer to the target. Ultimately, I was successful in creating this automated position feedback loop. However, the max intensity jumps around a lot, so one improvement could be using the center of the Gaussian fit to track the laser instead. For testing purposes, I used a low-power helium neon laser, but the next step is to apply my program to the high-power ytterbium laser in my lab. Thank you.
Hello, my name is Matthew Reilly. This summer I worked in the Yoon Biosensors lab. My project was centered around finding the best way to detect human papillomavirus, or HPV, using a smartphone, as it is the leading cause of oropharyngeal cancer in the US. To do this, we first amplified the DNA to make it detectable using RPA, which is an isothermal amplification method. We then confirmed that the amplification succeeded using gel electrophoresis, and once we confirmed that it was successful, we put the samples on microfluidic paper chips and recorded the flow rate of the samples through the chips using a smartphone. These videos were then analyzed and flow profiles were made based off of the flow rate. To find the best way to detect HPV DNA with a smartphone, we first used various amounts of cybergreen, which is a fluorescent DNA dye, and we found that 1 to 3 microliters of cybergreen worked best. Then we went on to our concentration testing, where we found that 10 to the negative 5 to 10 to the negative 2 micrograms per microliters of target DNA worked best. Finally, we combined our first 2 tests using various amounts of cybergreen and concentrations of target DNA and found that 2 microliters of cybergreen along with 10 to the negative 3 micrograms per microliter of cybergreen we're the best to detect HPV using a smartphone. Thank you.
Hi, my name is Ananya and I've worked in Dr. Egger's lab, which focuses on understanding the anatomy and physiology of retina under different conditions. 35% of people with diabetes develop diabetic retinopathy. Unfortunately, diabetic retinopathy is detected at a very late stage when most of the retinal damages is already permanent. The goal of this project was to look at the changes that take place in retina before permanent damages can take place. Specifically, I was looking at two layers of retina, the inner nuclear layer and the inner plexiform layer, and tried to see if there were any changes in the expression of CAV1.3, which is a calcium channel, on two types of neuron, GAD, GABAergic neuron, and BNOS, nitric oxide producing neuron. For this, we took retina slices and immunostained them with antibodies against the protein of interest, and then imaged them under a confocal microscope, which is a microscope with a high resolution. The area of the protein of interest and their overlaps were measured using ImageJ application. The results say that there was no significant difference in the expression of CAV1.3 on BNOS and GAD. This says that CAV1.3 does not change in early diabetic retinopathy, making it not a suitable target for early intervention or treatment. The lab will look at more slices to confirm findings and will perform more immunostainings for other calcium channels. Thank you.
Hello, my name is Enrique Sanchez, and this summer I worked in Dr. Barton's tissue optics lab. One of the focuses of this lab is the development of endoscopes for the early detection of ovarian cancer. This is important because ovarian cancer is the deadliest gynecological cancer due to a lack of early detection methods. Fortunately, studies have shown that ovarian cancer typically originates in the fallopian tubes as precancerous lesions. These lesions are present and detectable for many years prior to the development of ovarian cancer. To find these precancerous lesions, Dr. Barton's lab has developed a sub-millimeter cell-acquiring fallopian endoscope capable of capturing reflectance and fluorescence images of the fallopian tube. My project was to analyze images of human fallopian tube tissue with the goal of finding distinct signal differences between benign and cancerous tissue. Using different software tools, we used a mask to isolate the region of interest, grayscaled the image, and found the average signal intensity for each image. These ratios were then categorized by their pathologies, marking whether they're benign, cancerous, or precancerous. Throughout this project, we analyzed 28 fallopian tubes, with 25 being benign, 2 cancerous, and 1 precancerous. Due to the rarity of precancerous cases, a significant difference between benign and cancerous tissue cannot yet be concluded. Over the next year, over 100 more patient cases will be collected, and this data will be analyzed through the methods conducted here. Thank you.
Hello everyone, my name is Sham Satishan. I've been working in Dr. Gargeva's lab for genetically engineered model organisms and cell lines. I was tasked with finding out some specifics of a new method for genetic editing. This is based around CRISPR-Cas9, a system where an enzyme called Cas9 binds within guide RNA to create a double-stranded break in DNA. When the cell comes to repair the DNA, it can use a donor template. But if this donor template has a mutation, then that mutation will be reflected in the host genome. One way of delivering this donor DNA is through adeno-associated virus, a type of small virus that has transient properties, meaning it'll be disintegrated after delivering its vector. Traditionally, CRISPR reagents are delivered through lipofection, where they are surrounded by positively charged lipids to enter the cell. But viruses are typically delivered through electroporation, where electrical pulses create small pores in the cells. We want to find out which method was best for creating point mutations and entering cells. We conducted this experiment by designing and amplifying a plasmid vector, transfecting it into human kidney cells to create virus tagged with GFP, and then transfecting it into cancer cells by lipofection and electroporation. We then used flow cytometry, a method which uses lasers to detect photons to sort cells based on their viral presence. We then used PCR and a restriction digest for genotyping. We found that 140 cells could be sorted for virus, from liposuction, while only 57 could be sorted from electroporation. However, only 29% of the cells continue to grow from liposuction, whereas 49% continue to grow from electroporation. When genotyping, we found that 24% of cells had the desired mutation in the liposuction group, but 100% of the cells in the electroporation group had the desired mutation. This shows that electroporation is significantly more effective than liposuction and is worth exploring in the future. These results can help us determine whether electroporation is worth continuing in the future and how to improve on this, as well as exploring further research into certain types of research in creating models and treatments utilizing AAV's transient properties to create treatments for genetic diseases that can exit the body once delivered. Thank you.
Hi, my name is Corey Schneider. I was in Dr. Kai's lab during the summer. Our project was focused on anxiety and Prader-Willi syndrome, which Prader-Willi syndrome is a developmental disorder that causes excess anxiety. And there's a mutation on chromosome 15 that's associated with Prader-Willi syndrome. And so the mechanism of this disorder is not well known. So what we're doing is we did 2 tests to see exactly what's going on. The first test is an elevated plus maze, which assesses their fear of anxiety by having open spaces and height. And also we did an open maze just to see their fear of open spaces. So a normal mice would be scared of the open spaces and spend more time in the closed arm. And we could quantify that by using a tracking data software to see how much time they spend in the open versus closed arms and the distance traveled between those arms. And so if we look at our data, we can see that the control mice, which is the regular mice in this experiment, did spend more time in the closed arms as expected, showing that they were more scared. And the open maze does confirm the results by being consistent. But the MIGL2, which is the mice with the Prader-Willi syndrome, are less scared for some reason. They show less anxiety, and they spent more time in the open arms and traveled more in the open arms, and the open maze also is consistent with the results. This suggests that Prader-Willi syndrome is a more complex disorder with the anxiety part of it, suggesting that maybe the MIGL2 has something more than we expect. So the further research direction would be to analyze the image in the brain to see exactly what's going on at that time in the central amygdala, which is the fear center of the brain. Thank you.
Hello, my name is AJ Schaefer, and this summer I worked in Dr. Pappas' lab studying cellular therapies for treating type 1 diabetes using insulin-producing cell pouches that do not require immunosuppression. Type 1 diabetes is a chronic autoimmune disorder that affects 8.4 million people worldwide, including myself, and stems from the body attacking beta cells in the islet region of the pancreas. Beta cells are insulin-producing cells found in islets, which are cell clusters that regulate the body's blood glucose. While islet transplantation is a current form of treatment for type 1 diabetes, it requires patients to take immunosuppressant drugs, making it not widely applicable due to factors like age, pre-existing conditions, and compromised immune systems. Encasing islets in something called a cell pouch with an immunosuppressant membrane makes this treatment widely applicable and allows for people to have this treatment without the use of immunosuppression drugs. The focus of my study was looking at the beta cell composition, or insulin to islet area, after prolonged encapsulation to see if these islets survive in these encapsulation chambers. To do so, I took an IHC, or immunohistochemistry, fluorescent stain with slides from 1, 2, 7, and 14 weeks taken from rats, and I used primary antibodies that bind to insulin, which fluoresces green, and that bind to glucagon, which fluoresces red. I also used a DAPI antibody, which binds to cell nuclei fluorescing in blue. What I found is that after 14 weeks, or prolonged encapsulation, islets maintained a similar beta cell composition, or insulin islet area, when compared to islets in culture and islets from earlier encapsulation periods, such as 1, 2, or 7 weeks. What this shows is that this is a proof of concept for the effectiveness of treatment using islet transplantation in these cell devices, and this opens the door for future research involving human stem cell-derived islets, which offer a virtually unlimited supply of insulin-producing cells for future cell transplantation and possibly clinical trials for treating type 1 diabetes in the future. Thank you.
Hi, my name is Jalen Sherfield, and I work in Dr. Walty Lab, where we focus on malate dehydrogenase and its possible interactions with PFAS. Malate dehydrogenase is a protein in the mitochondria that deals with ATP synthesis, and when it malfunctions can cause disease like cancer and brain malfunction. PFAS are industrial chemicals that have become recent cause for concern due to their toxic and resilient nature, and we wanted to find out if and how these PFAS chemicals interact and possibly cause malfunction of malate dehydrogenase. How we did this was, first we designed a plasmid that codes for MDH, or malate dehydrogenase, and then we inserted it into E. coli and grew the E. coli to produce the MDH in abundance. Then we filtered out our MDH with various methods, including FPLC. Then we introduced our PFAS, in this case PFOA. Then we ran an NMR titration spectra, which measures different structures of molecules using magnetic interaction at different concentrations to estimate binding and interaction between molecules. According to our results, we were able to successfully isolate our MDH protein, and our NMR spectra shows that there is binding between MDH and our PFOA, and there is also possible complex formation between the two. In the future, we plan on using cryo-electron microscopy in order to visualize this binding instead of just confirming it, and we also plan to use different PFAS and different mitochondrial proteins. Thank you.
Hello everyone, my name is Inara Smith, and this summer I worked in the O'Hare lab under Dr. Sarah Layton. Primarily, they are currently focusing on service dogs and their impact on individuals with disabilities. So, are they raising quality of life? However, while service dogs are extremely important, they are also lacking in a standardization for their training and research. So, this is what my project is focused on. My first aim was to acquire accurate data on key metrics around service dog training, such as cost, time to train, wait time, etc. My second aim was to find out if the role of a dog has an impact on their cost. My method was through 3 accrediting organization websites. I compiled a list of 91 organizations in an Excel sheet. Then, through individual searches from websites, Google, things like that, I acquired the data I needed in that Excel sheet. Then, I put those numbers through R, which gave me these results. If you look at the table in the bottom left, you will see my first aim depicted. This shows you the average, the minimum, and the maximum for each of the key metrics that I compiled. In the top, you will see a table that shows you the difference between each service dog role and their cost between different organizations. On the bottom right, that is a visual representation. The first table I had you look at will show you the service dog industry does have a lack of standardization. Wait time can range from 0 to 36 months. Costs vary from 0 to upwards of $20,000. There is a lot of variability in the industry. From the top table and the bottom right graph, you will see that service dog role generally does not have an impact on their cost. They all hover around $40,000. In the future, these metrics can be used to be cited in sources in relation to the lack of standardization in the service dog industry. Thank you.
Hi, I'm Jackie Stewart, and this summer I worked in Dr. Kong's material synthesis lab. Magnets are essential in modern technology ranging from electric vehicles to wind turbines to consumer electronics. However, they are dependent on a class of elements found near the bottom of the periodic table called rare earth elements, which are problematic because they are detrimental to the environment, expensive, and limited in supply. That's why our lab is working to replace them with transition metals, the block of elements in the middle of the periodic table, because often their unpaired electron spins can lead to strong magnetization. I'm working specifically with 2 compounds, strontium niobium oxide doped with some titanium, as well as hafnium iron silicon, and investigating how changing their elemental ratios could change their magnetic properties. First, after making the compounds, I wanted to homogenize their composition and structure by heating them up, so I put them in either a small box furnace or an arc melter. Then, I wanted to verify that I formed the compounds that I actually wanted to by putting them in an X-ray diffraction machine. Finally, I measured their magnetic properties in a vibrating sample magnetometer. So here's what we found. Although we don't have enough space to show all the data, for all ratios of titanium in the first compound and lower ratios of silicon in the second compound, we found that our compounds were pure because the patterns of our reference data matched the patterns of our own data, as seen in Figure 7 and 8. However, in Figure 9, that was not the case, because the green, showing a high concentration of silicon, had extra peaks compared to the black line, which shows low silicon concentration. That is why we decided not to measure magnetization data for our silicon compounds, and instead only for titanium. If we look at Figure 10, we see that the blue has no titanium and the orange line has some titanium. And across, for both of them, we see that magnetization did not decrease significantly as we increased to room temperature, which is good. However, our magnetization was negative and also really small. Although that was unexpected, we did find that mixing more titanium did increase the magnetization and make it closer to the positive range, which is what we wanted. In the future, we should better define the stability of our silicon compounds and mix in more titanium to see if that would make our magnetization significantly stronger. Thank you.
Hi, my name is Uma Isa Urias, and during my summer, I worked in Bob Koseka's Plant Soil Environment Lab, where we studied the elemental components within plant species. We know that plants are capable of accumulating certain metals, and plants that accumulate metals 100 to 1,000 times more than the majority are known as hyperaccumulators. We hypothesized whether plants within the same species share identical elemental compositions to one another, even with differing geographical locations. To determine this, we took species under the plant families of Nokia and Thalaspi and studied their metallic aspects. We did so by using a special X-ray scanner called the PXRF scanner, which is able to detect every elemental measurement within each species being scanned. Once we collected our data from the PXRF scanner, we then organized it into an Excel spreadsheet and created box plots to see if we could visualize any major differences or patterns within our data set. The most statistically significant difference we had seen were between the elements of nickel and zinc, where I am proud to say that one of the hyperaccumulators I discovered accumulated more than 7,000 ppm of zinc, which is now the highest ever recorded level of zinc accumulated from a species in the Nokia plant family in all of the United States. From these results, we can conclude that even when a species is known for hyperaccumulating certain elements, this does not mean that every plant within the species will share the exact same elemental components to one another. Studying how plants accumulate metals is important when understanding how environmental traits can affect the overall process of accumulation, and can lead to a further discussion of taking these plants to process them and extract these metals to utilize. Not only will this provide a greener way of mining that does not destroy our surrounding environment, but this also aids the U.S. in self-providing in important domestic resources, supporting our country's self-efficiency as a whole. Thank you.
Hi there. My name is Matthew Weisberg. During my summer, I worked in Dr. Churko's heart muscle lab. In this lab, we worked to aid in the discovery of heart arrhythmia therapies. Heart arrhythmias are when there's an irregular rhythm in the heart cycle. This is very dangerous because it can lead to sudden strokes and sudden death due to inadequate blood flow. Up to 60 million people worldwide are affected by certain types of heart arrhythmias, making them very common and dangerous. My job in Dr. Churko's lab was to grow heart muscle cells that we took stem cells from patients who had diseases. We would then take these heart muscle cells and add drugs in them in order to test the rate of contraction using videos. These videos were then used with home scripts to take data for future analysis and eventual drug discoveries and drug use in the future. Thank you.
Hello, I'm Cole Younger, and this summer I worked in the Largent Milnes lab, which studies the physiology behind headache and migraine. For context, headache and migraine are common debilitating conditions that stem from a dysregulation of the endocannabinoid system, the brain's pain modulation pathway. The periaqueductal gray, or PAG, is a vital component of this pathway that influences headache initiation and persistence. Furthermore, astrocytes, regulatory cells within the brain, are found to play an active role in sustained neuroinflammation associated with headache. Previously, my lab developed a protocol to isolate and culture astrocytes from mice spinal cords. This summer, I adapted this protocol in order to isolate and culture astrocytes from the PAG of mice, which will allow us to study these astrocytes' behavior and health in vitro or within a controlled environment. Additionally, we investigated CB2, a vital endocannabinoid receptor, through both behavioral testing and astrocyte cultures of CB2 lacking and typical mice. Through our first isolation, we found that 1 PAG is sufficient for cell cultures. Then, through behavioral testing, we discovered that CB2 is vital for pain management in female mice. Finally, by applying our protocol, we compared typical and CB2 lacking astrocytes through both cell quantification and imaging. Through imaging, we found that the typical astrocytes were more developed. However, the adherence of microbeads to cell surfaces hindered exact quantification and imaging. Ultimately, our experiments illustrate that our cultures can be used to compare astrocytes across genotypes, and in the context of migraine and headache research, our cultures can be used to investigate the effects of medication overuse headache-inducing drugs such as opioids and triptans and how they affect astrocyte health and behavior. Thank you.
Hi, my name is Dong Yu, and I worked at Keys this summer in the lab of Dr. Srihan Aras. My project focused on analyzing heart rate variability, or HRV, to study stress changes in young adult cancer survivors before and after mental health interventions. Chronic stress can persist long after cancer treatment ends and negatively impact recovery. Traditional surveys can help assess emotional well-being, but they often miss the real-time physiological changes. To address this, we used Empatica E4, a clinical grade sensor that tracks heart rate and other signals continuously, especially during sleep when data is less affected by daily activity. We focused on the interbeat interval data, which captures the time between heartbeats. We cleaned and filtered the data in Python to remove noise, outliers, and other physiologically implausible values using custom functions. Next, we segmented the data into 5-minute and 60-minute time windows to optimize calculations for various HRV metrics. To make our findings interpretable, we visualized the changes over time using distribution graphs and bar plots, allowing us to compare HRV trends before and after interventions. Our results showed a decreased low frequency to high frequency ratio in 7 participants, indicating a more relaxed bodily state. RMSSD increased in 4 participants, which indicates increased short-term recovery and decreased overall stress. However, it's worth noting that RMSSD can be affected by respiratory artifacts. Overall, we saw modest but meaningful improvements in various stress-related physiological signals. Although the trends are subtle, they show that wearable data can reveal real-time changes that traditional surveys might miss. This kind of analysis has the potential to support personalized, data-driven care by helping clinicians evaluate the effectiveness of interventions objectively. Thank you.
Hi, my name is Megan Zhu, and this summer I worked in Dr. Schwartz's cancer research lab. Specifically, my mentor and I worked on Ewing sarcoma, which is a type of bone cancer that's prevalent in young people and adolescents. Ewing sarcoma is especially hard to target because of its aggressive nature, drug resistance, and rapid spread. Now, G quadruplex is an abnormal 4-stranded DNA that's thought to play a key role in Ewing sarcoma. Based off this, we hypothesized that drugs that target and unravel G quadruplex can effectively kill Ewing sarcoma. We also wanted to find drug compounds that solely targeted Ewing sarcoma cells with minimal damage to healthy cells. We tested this by making 2 different cell cultures of both cancerous A673 cells and non-cancerous HEK293 cells, and sending them to a lab to be tested with 4 different drug compounds. Our results showed that all drug compounds effectively killed 50% of the cancerous A673 cells, with compound 7 being the most effective. Figure 5 compares all drug compounds on both cell lines, showing that each drug compound effectively killed more Ewing sarcoma cells than healthy cells, except for compound 40. In future research, DHX9 plasmid can be transfected into Ewing sarcoma cells to check our hypothesis and the effectiveness of these drugs. This can allow future researchers to develop a more concise drug that will solely target Ewing sarcoma cells with minimal damage to healthy cells. Thank you.
Hello, my name is Akai Shabari, and I worked with Dr. Manalini Kala in the Department of Internal Medicine and Dr. Taban Hale in the Department of Basic Medical Sciences. Cardiovascular disease is the leading cause of death in the world. One of the causes leading to heart failure is high blood pressure, and this is linked to how heart cells become overly active, and this can eventually result in fibrosis. Angiotensin II acting at the type 1 receptor, otherwise known as the AT1AR gene, can lead to hypertension and cardiac fibroblast activation. Cardiac fibrosis occurs when there's an overdevelopment of scar tissue in the heart due to cardiac fibroblast activation and replication. Cardiac fibroblasts are cells in the heart that create scar tissue. Prior studies have shown that there are sex differences in the number of cardiac fibroblasts and how cardiac fibrosis develops. In addition, blocking the AT1AR in males has been shown to reduce cardiac fibroblast number in the hypertensive heart and reduce fibrosis. So we investigated how deletion of the AT1AR, specifically in cardiac fibroblasts, influences cardiac fibroblast number in male and female mice. First, we isolated the left ventricle from heart tissue of 6 male and female mice each, and then we analyzed cardiac fibroblast number through flow cytometry. The readout for the cardiac fibroblast is the fluorescent protein T tomato, which is only found in these cells. Our findings showed there is a higher proportion of cardiac fibroblasts in female mice, but deleting the AT1AR gene did not appear to impact overall cell proportion. Understanding sex differences in cardiac fibroblast biology may lead to more optimized treatments, such as targeted therapies for males and females experiencing cardiac fibrosis. Thank you.
Hello, my name is Derek, and I'm a senior at ACP. I'm a virtual intern working in Dr. Lee's computational biology lab. I researched into whether object detection models such as DETR and Detectron-2 can distinguish spinach plants from unwanted weeds under diverse field conditions. Spinach is unique in that it looks extremely similar to weeds when it is a seedling. With an accurate object detection model, targeted weed solutions can be implemented to reduce herbicide usage and optimize crop yields, enabling sustainable agriculture. Object detection models, put simply, work by learning input data patterns and predicting where objects are and what they are. By creating over 7,000 box annotations across 123 images, along with weed data provided by my lab, I trained DETR, an end-to-end object detection model, and Detectron-2, a modular framework for object detection models, and I found that Detectron-2's average precision and average recall scores were significantly higher than those of DETR. DETR had high confidence, but had many false spinach detections. This was likely because spinach often grows in dense regions, leading to overlap. With high accuracies, object detection models serve as the foundation for all proposed targeted weed solutions, whether it be automated spot spraying or laser reading. Thank you.
Hi, my name is Shrishti Chaudhary, and this summer I worked in Dr. LaFleur's biostatistics lab, where I looked at cognitive health and aging. So my project is also part of the Precision Aging Network, which is a collaborative project that focuses on normal aging given age-related health issues. So my study specifically looked at 2 cognitive tests. The first one being the Paired Associates Learning Test, or PAL, which focuses on visual memory and learning. The second one being the Keep Track task, which focuses on executive function. My project also had 2 research questions. The first question being if PAL and Keep Track tasks are correlated with Paired Associates test, which is another baseline cognitive test, and if these associations differ in people who have cognitive impairment versus people who don't. To answer this question, we used a method called analysis of covariance, which allowed me to compare these test scores with the cognitive status. My second research question looked at the Keep Track task and PAL and if they were related to demographic data such as health, sex, BMI, and hypertension. To answer this question, we used a method called multiple linear regression. Using this, we were able to compare the health factors with the test scores and see if they were correlated. The results showed that mild cognitive impairment was visible with people who had lower test scores in both PAL and Keep Track. We also saw that older adults and people with higher BMI had lower test scores, and females had higher test scores than males. Overall, tests that focus on memory and executive function are more sensitive in measuring cognitive function in healthy aging adults. Thank you.
Hi, my name's Kapil, and this summer I worked in Dr. Chen's lab, focusing on investigating functional MRI methods and their applications to studies of the glymphatic system. More specifically, I looked at how cerebrospinal fluid was flowing throughout the brain, which is important when researching diseases such as Alzheimer's. To start, I analyzed 4D MRI and fMRI data and in 3 different manners. I computed the standard deviation, performed temporal analysis, and calculated the spatial correlation. Using standard deviation, I was able to see how cerebrospinal fluid was flowing throughout the brain and analyze the glymphatic activity. Using temporal analysis, I was able to see which areas of the brain were being activated during a finger tapping task. And lastly, using spatial correlation, I was able to see if having greater amounts of CSF in an area of the brain was correlated with greater brain activations in those specific areas. Looking at figure B, we can see an axial MRI slice of the brain. In the middle, we can see 2 streaks of white representing the cerebrospinal fluid flow. Moving to figure C, we can see red dots annotated on the sides of the brain. Higher concentration of red dot in this image represents greater brain activation during the finger tapping task. Using both of these results, I was able to derive a spatial correlation coefficient of 0.0698, signifying that there isn't much of a correlation between having increased CSF and brain activation in those areas. Moving forward in the future, this number and analyzing the spatial correlation coefficient is important because it can provide insight into treatments and further research into Alzheimer's and other neurodegenerative diseases. Thank you.
Hello, my name is Ayana Jane, and this summer I worked in Dr. Isabel Schroen's lab, which mainly focuses on developmental disorders in children. As such, my project looked at extreme and rare inner ear and auditory nerve malformations, and my goal was to find causal variants. These disorders and syndromes have many unknowns, so finding these causal variants allows for further understanding and possible treatments in the future. My project had 2 parts: computational analysis and wet lab sequencing. We began with verifying reported family relations and then moved on to looking for mutations in the genetic data of the family and the children. Once finding these mutations, we eliminated them based on frequency in the population and then looked to determine whether or not they could be pathogenic or dangerous to the ear. We did this in 2 ways. We looked at something called the CAD score, which determines how dangerous in general the variant could be to the body. The higher the score, the more dangerous. We also looked at G-ear, a database which shows how highly expressed a gene is in the ear. If the variant was found to have both of these conditions, we moved on to wet lab sequencing. We conducted 3 tests: PCR or polymerase chain reaction, gel electrophoresis, and Sanger sequencing. These tests amplified and separated the DNA and then ensured that mutations actually existed in the DNA by verifying the base pair order. After doing this, we found 7 mutations after computational analysis, and 5 were confirmed by Sanger sequencing. 4 of these mutations were a type of variant called compound heterozygous. This means that each parent carries a different mutation on the same gene. We found compound heterozygous variants on the FRAS1 gene and the PIT-RM1 gene. These variants can now be uploaded to something called GeneMatcher. GeneMatcher is a database where we can upload families, their genetic DNA, and the variants we found, and we can see if other families had the same variant and the same symptoms, therefore ensuring that this variant is pathogenic to the ear, causes this malformation, and then in the future we can look for possible treatments. Thank you.
Hi, my name is Audie Giulicanti, and over the summer I was in Dr. Shireen Durgar's lab in the Translational Cardiovascular Research Center, where I focused on the role of a protein in the heart called PAM. Heart disease is the leading cause of death in America, so by being able to analyze how PAM functions, we can manipulate it in such a way where it becomes therapeutic to us when we have heart disease, or more specifically, cardiac hypertrophy, which is the enlargement of the heart muscle. With that being said, we investigated the role of PAM in cell size and gene expression to see if PAM could act as an anti-hypertrophic agent, which would prevent the enlargement of heart muscle. We did this because in our past research, we saw that PAM on the transcription level increased significantly when there was applied pressure to the heart. To start our research, we eliminated the expression of PAM in half of our infant rat cells and compared it to the other half where PAM was expressed. We did this for gene expression and cell size. For gene expression, we performed cDNA synthesis where we isolated the RNA and reverted it back into DNA to see what genes were expressed. We saw that when PAM was eliminated, endoplasmic reticulum stress genes such as CHOP or HSPA5 increased significantly. For cell size, we used a phase contrast microscope to take pictures of cells that had PAM and did not have PAM. We saw that cells that did not have PAM were much larger due to becoming hypoxic, which means a lack of oxygen. This would lead to cell death. Our data shows that PAM can indeed act as an anti-hypertrophic agent, as it has a vital role in gene expression and cell size. This can be used for future clinical research in the future. Thank you.
Hi, my name is Anvira Kerr, and this summer in KEYS 2025, I worked with Dr. Tyson Swetnam in his Artificial Intelligence lab. We looked at the improvements in research that could be done by integrating artificial intelligence into any researcher's workflow. We were determined to prove that LLMs, or large language models, integrated with the new and upcoming concept of MCPs, Model Context Protocol, would make a researcher's work more efficient and less time-consuming. MCPs can access databases from a user's computer and any external applications that are needed for the researchers to conduct their research. This makes it less time-consuming and sets up a workspace for a researcher that takes a lot less time and more efficiency. As you can see from my materials and methods, before MCPs, the process of research was much more time-consuming. Researchers had to collect their own data, put it into databases, organize it, and analyze it themselves using external applications. But after MCPs, this process got so much more streamlined. Researchers could just access an LLM-integrated application or an application that has AI in it and have their presentable data within just a matter of minutes. I chose to show my results by creating a chatbot for my website. This is a great example of efficiency, since users that access my website can just go prompt the chatbot and get any information they want about my website without having to manually search its pages. At the end, these findings may revolutionize research by creating powerful interconnected environments where scientists can change the way they talk to AI, and maybe AI can be useful too. Thank you.
Hi, my name is Kenji, and I worked under Dr. Ryan Gutenkutz's lab for biotechnology and statistics, and I researched about artificial genomes and its extent on research. One of the important things about artificial genomes is that it can mask people's actual genomes so that it is not compromised when used in research. To make an artificial genome, we used a thing that's called Generative Adversarial Network, or GAN for short, which has two steps. One is the neural network step, and the other is the learning algorithm. The first neural network is simulating what a human brain would look like. Based off of patterns of input data, we are able to create generative genomes. The next step is the learning algorithm. Based off of those generated genomes in the neural network, it can compare with the real data to see if it is and does mimic an actual genome. What we found in this process was by running it through a software called DADDY, made by Dr. Ryan Gutenkutz, we are able to find that the demographic inference, or the population size, based off of NU versus time graph, we can see that the shapes of the artificial and the real genomes are not similar. This concludes that we are unable to similarize the artificial genomes with the real genomes as well. Based off the NU and time graph, we are unable to use the artificial genomes in research because it does not mimic the real genomes as well. This means that artificial genomes in this research purpose for population dynamics, we are unable to use artificial genomes to replace real genomes. Thank you.
Hi, my name is Hannah Liu, and this summer I was working in Dr. Maisel's astrobiology lab, where they look at early life and the genetic code. I was focused on one specific protein, reverse gyrase, which is found in organisms living in extremely hot conditions. It supercoils the DNA in these organisms to keep it from falling apart.
The origins of this protein are highly debated, so my goal was to better understand the origins and evolutions of the reverse gyrase protein and determine whether or not it was found in the last universal common ancestor of all existing life. To do this, I looked at the specific units within the reverse gyrase protein, downloading protein domains associated with reverse gyrase and looking at the most common ones to form a hypothesis on how reverse gyrase came to be. Then I made a gene tree where I could trace reverse gyrase's evolution through bacteria and archaea. My results showed me that reverse gyrase is made up of 4 main protein domains. With these 4 main protein domains, different combinations of them were constructed and compared with architectures in other proteins. Through this, I hypothesized how exactly the 4-domain reverse gyrase came to be through a series of events. For my gene tree, it showed me that reverse gyrase likely evolved through horizontal gene transfer between and in archaea and bacteria between organisms rather than originating in a single common ancestor, meaning that it likely was not found in the last universal common ancestor, and this shows us a little bit more about early life and the conditions in which it lived. Thank you.
Hi, my name is Ishan Ranjan, and I worked under Dr. Bonnie LaFleur's Precision Aging Network Lab. To understand my project, there are many different types of ages. I specifically looked at 2, chronologic age and epigenetic age. Chronologic age is your calendar age, or the amount of birthdays you've had. Epigenetic age is your biologic age, and essentially specifically tailored to your DNA. To understand this, think of your favorite Hollywood actor or actress. They look like they're 20, but in reality, they're pushing 50. That's epigenetic aging in action. Specifically for my project, I looked at how vitamin D, a potential anti-aging factor, could slow down epigenetic aging and turn back your biological clock. I also had a sub-question of looking at phenotypic associations with epigenetic age, including but not limited to sex, BMI, education, etc. The motivations for my project were 2 different studies. The first was VITAL, which found out that vitamin D supplementation will decrease your epigenetic age by 3 calendar years. Additionally, was Do Health. Do Health found that vitamin D plus omega-3 fatty acids will decrease your epigenetic age. However, vitamin D alone will not. So for my project, I looked at Precision Aging Network data and tried to see if I could verify that. For the Precision Aging Network data, I had to first find the correlations between epigenetic clocks, things that look at your epigenetic age versus your chronological age. Here I found the first generation clocks from the PAN data were similar to the second generation clocks from the Do Health, and the third generation clocks from both were identical. Now, onto my results. For the Horvath clock results on the left side, you see that sex is statistically associated with a lower epigenetic age, where females have a lower epigenetic age than males. I guess the males need to step it up. Vitamin D, however, did not have a statistical correlation. Now, onto the Dundon-Pace clock graph. Here we see the same statistical association with sexes. And then for education, we see as the amount of education in years increases, your epigenetic age decreases. And we see as your BMI increases, your epigenetic age will actually increase. And we see the same inconclusive result for vitamin D. The reason for the inconclusivity of vitamin D is that the PAN data didn't take into account how long the users had taken vitamin D or the amount they had taken. VITAL and Do Health did take this into account. So for future studies, the PAN data should look at the duration of supplementation. Thank you.
Hello, my name is Shivani Satish Kumar, and this summer I've been working in Dr. Wooden's lab under the mentorship of Dr. Pam Miranda to learn about the lateral habenula. The lateral habenula is a part of the brain that regulates reward-seeking behavior, kind of like a break of when to stop. We've been looking at specifically how this part of the brain reacts when posed with a threat. To do so, we performed the looming test. The looming test is an expanding black disc which simulates an aerial predator attack. Before doing the looming test itself, we used viral injection brain surgery to fully visualize the results. We then habituated the mice in order to fully assimilate it to its environment and eliminate any other possible stimuli. Lastly, after the looming test, we used CFO staining to quantify the amount of cells activated throughout the test and input areas. From this, we performed 3 types of results. During the habituation stages, we used the open field test to establish a baseline of behavior. We found that all the locomotor activity within all the mice were similar. Through the looming test, we found it to be successful as it evoked a response out of the mice. We found an increase in velocity after the looming presentation and saw that the mice were more prone to running towards the shelter. Lastly, we're still in the process of quantifying the amount of cells through CFOs. However, we expect to see an increase in the lateral habenula and are examining which areas inputted towards this final decision. We hope that future research helps create treatments towards this area and seeking behaviors. Thank you.
Hello, my name is Saish Vamadure, and I worked in Dr. Nirav Merchant's lab to create Converse with Your Data, a full-stack application that allows researchers or people for recreational use to use data analysis without knowing any coding language. This allows them to create SQL translations, which is a type of coding language that allows them to interact with databases and allows them to visualize the data without knowing any Python. The materials and methods I used for this full-stack web application was first Psi versus Verde AI. This gives me access to Llama 4 Scout, an LLM, or large language model that allows me to do a lot of the back-end processing. One key example of this in my project is SQL translation. A human can put in an English prompt or any other language, and then the LLM in the back-end will translate this into SQL and run it against the database, which then results with the SQL that was intended and the desired output. Next, this also suggests visualizations for the user. Second was Plotly. Plotly is a Python library that allows me to create visualizations super easily with inbuilt functions and allows for the visualization part of the application. And third is Docker. This allows me to make the data reproducible and make the software reproducible by putting all of it and its dependencies, i.e. Plotly for example, into one file that users can easily put onto their computer. Next is the results. As you can see, the first introduction page asks for the table name, the data itself, and then the line it starts at to make it easier for the system to read the data. Next, on the second page, it does three things. One, it suggests visualizations for the reader. You can choose, for example, histogram or bar chart, and it'll output that for you. On the top right, it has the embedded data file itself for the user to look through it, and then on the left, it allows the user to write human prompts and get the SQL and the translation. Thank you.
Hello everyone, my name is Ryan Tom and I work under Dr. Guan Yao's lab at the University of Arizona for my project. Ovarian cancer remains one of the most lethal forms of cancer that can be contracted by women. It is primarily caused by a mutation in the BRCA gene, a gene which is notorious for causing breast cancer as well. MicroRNA regulates the expression of the BRCA gene, or how much of an effect it has. So my project was to determine what the correlation is between the number of microRNA in a patient and their likelihood of developing ovarian cancer. To do this, I used a support vector machine. A support vector machine is a form of machine learning that extracts patterns from a training dataset. It uses those patterns on unidentified points and classifies them into two binary categories, which are provided by the training dataset. In my case, I had low and high risk as my categories. I trained the support vector machine and then I had to tune it. I tuned two parameters, cost and degree. Degree is a parameter of the support vector machine that transforms data into different dimensions depending on what the number is. It allows the support vector machine to find patterns that it may not have found in smaller dimensions. Cost is the support vector machine's tolerance to misclassification. I tuned both parameters with multiple different values, and I ended up finding that 1 degree and a cost value of 1 EO6 were the best values for the model. All in all, I got an accuracy of 92%. This model can be used by physicians to create care plans prior to a patient fully developing ovarian cancer. Thank you.
Hi everyone, my name is Aditya Tyagi, and this summer I worked in Dr. Hale's lab at the University of Arizona College of Medicine, Phoenix. Now, for my project, I learned about how angiotensin II, which is a molecule often known for causing an increase in blood pressure, can often lead to inflammation in the heart. Now, often for people with high blood pressure, inflammation can damage heart tissue over time, eventually leading to heart disease. Now, in order to study this, we use an in vitro and an in vivo model. For the in vitro model, I treated rat2 fibroblast cells with angiotensin II in a dose-dependent manner for about 24 hours. I then used an ELISA, which is an enzyme-linked immunosorbent assay, to detect for the amount of IL-6 release into the media, which is an inflammatory cytokine. In addition to that, I used Western blotting to detect for NOX2 expression, which is basically a type of enzyme that causes oxidative stress and also activates inflammatory processes. Now, for the in vivo treatment, my lab cultured and treated male spontaneously hypertensive rats with angiotensin II for about 14 days. After that, I used ED1, which is a type of macrophage marker, to detect for macrophage infiltration in these male rats using immunohistochemistry. Now, both of these in vitro and in vivo models have showed us both the whole body and cell-specific functions of the treatment response to angiotensin II. Now, as for the results, we found out that angiotensin II can actually lead to an increase in IL-6 production as well as an increase in NOX2 expression in a dose-dependent manner. We also noticed that there is an increase in immune cell infiltration in the rat heart tissues. Now, all of these results have led us to believe that angiotensin II can actually promote inflammation and cause oxidative stress, which are all key factors that contribute to heart damage. Thank you.